Thank you, Stephen. It's a pleasure to be here and see so many familiar faces. So it's happy to talk to you a little bit more about Arbor Bio. We are a next-generation gene editing company, and I'll tell you about the way we're thinking about the editing space. So Arbor was co-founded about 6 years ago by Feng Zhang, who's the CRISPR pioneer from the Broad, and David Walt, who had co-founded Illumina and Quanterix. So David has an expertise in high-throughput automation, and Feng obviously in CRISPR. And the company was unique in that it was not founded on any technology that came out of the Broad or Harvard. It was actually founded on an idea. And the idea was that Cas9 was not the best nuclease found. It was just the first one that worked. And if we were to go out and look across the metagenome, would we identify better, more differentiated approaches to editing beyond Cas9? So that was kind of the founding ethos for the company. So the company built a very strong machine learning AI-driven discovery platform to enable us to quickly identify novel editing technologies, engineer them, and get them ready for putative therapeutic use. Flash forward now, we now have what we believe is the most extensive toolbox of proprietary gene editing approaches, which allows us to really focus in on what's the underlying disease pathology we're trying to fix, and then bring the right tools to bear. We're focusing in four areas around sort of genetic disease. I'll tell you a little bit about that. And the other important piece, I think, is making sure we have the right team in place. So we do have a very experienced team that has achieved over 20 INDs in the cell gene therapy gene editing space, which is important for that translation. And from a pipeline perspective, we are focused in on really a stepwise risk mitigation approach. So we have a lead or gateway indication for each of our technology platforms, and upon success, we can then branch out more quickly. We are focused in the liver and CNS and our targeted first filings in the next 9 to 12 months. And it would be the first non-Cas9 tested in vivo in humans. We've raised over $300 million to date. Our Series B was in November of 2021 for $215 million, co-led by Temasek, TCG, and Alley-Bridge Group. And Arch Ventures was the original lead on the Series A. We have multiple partnerships, and I'll mention a little bit about those later. So as I mentioned, what we really want to do is understand that underlying disease biology first, determine what do we need to affect from an editing perspective, and then pull the right element from our toolbox to really enable us to treat and bring significant benefits to the patient. Now, if you think about the world of what you can do in genetic diseases, there's kind of four big things you can do. One is to knock something down. There's a protein that you do not want there. You want to just eliminate it. And so that's a technology we have the ability to do. There's also diseases where you have too much of something and you need to excise a precise piece. And so a precise excision, that's another core focus area of ours. A third area is being able to rewrite or replace smaller sections of DNA from SNPs up to, say, 100 base pairs. And we utilize reverse transcriptase-based editing for that approach. And then finally, the fourth big bucket is being able to drop in entire exons or whole genes. And for us, it's going into the endogenous loci. We're not taking a safe harbor approach at this point. We feel like the endogenous loci, we're actually under endogenous controls where we want to be, where we have less worry about therapeutic indices because it's under the right control systems. We're also not doing base editing at this time because we feel RT editing enables us to have a little more flexibility in how we think about rewriting sections of DNA. So these are kind of the four areas, and it's underpinned by making sure the key technology across each of these is having a nuclease or some type of nuclease approach, whether it's a double-strand cutter, a nuclease, or some other in-between approach that allows us to really touch anywhere in the DNA we want. So by having a portfolio of very small nucleases, a portfolio of nucleases that have a variety of sites that tell them where to bind on the DNA is critical. And then finally, a variety of nucleases that also have different cut types. So you can have blunt cutters, you can have staggered cutters. Again, it all depends on what you need to do to affect treatment for that patient. And so with that stable of nucleases and the variety of PAMs and sizes, it allows us to really think about how can we differentiate in each of these areas. And that's an important piece for us as we think about both diseases we target as well as the technology approach to make sure we can do something that today others cannot or are unable to do. We don't feel the need to be a third or fourth in a particular space. It's important to be the first utilizing a very novel approach that's differentiated. And so you can see across these, I won't go into the details here, but having very small effectors makes a difference. Having a variety of PAMs enable us to get adjacent to the site we need to edit, which is really important. And then finally, being able to really work out how do you do this efficiently, for example, with nuclease excisions, so that you can actually keep the open reading frame intact if you're doing a particular excision. So once we've outlined sort of the technology focus areas, how do we want to think about the lead indications? And again, the way we think about it is each one of these has a very different risk-reward profile. And so to really mitigate that lead indication, you want to de-risk everything around it except for your core technology. So that means you need to make sure your indication you select has validated biology, there's a clear clinical and regulatory path, and there's a clear approach for delivery. And oftentimes that means you're going to start with an orphan disease that allows you where you can have much tighter control over these elements. And so that allows us to make sure for each of these technologies we select one indication that really fits the bill, that we can really optimize the technology to that. Upon success, you can then expand that platform. Where we're focused today, you can see across the different technology platforms, we've got a variety of approaches. And we are only focusing on in vivo. We partner on the ex vivo space to allow folks to have access to our technology, but we are not going to develop our own ex vivo cell therapies. That is on purpose. There's very little synergies for all of you who know this in the space between a cell therapy and a in vivo genetic medicine. And so let's focus on one thing and do it well. And so we're focusing just on the in vivo side, partnering ex vivo. Across each of these, the program I'll tell you a little bit about today is our lead program in the knockdown plus space, which is primary hyperoxaluria, which is our lead program. And you can see we've got a gateway program for each of our technologies. Delivery is another key element for all of us. And as we think about liver, LNP delivery, I think, is relatively well validated. We've seen success with some of our peer companies like Intellia and others who have really demonstrated they can deliver to the liver and see successful data in humans. So we're utilizing LNPs for the liver indications. Our initial, we've done a partnership with Acuitas to access their LNP technology. Outside of liver, I think it's a little bit, remains to be seen. I think there are AAV approaches that allow you to get to specific tissues, and there's a lot of work in non-viral delivery outside of the liver ongoing. Much of it quite early, but at some point it'll break. So for our CNS indications, initially we're focused on utilizing AAV, where we can get to specific cells for specific diseases where we can do the editing. So primary hyperoxaluria is a life-threatening condition. In this particular, in patients, they have a mutation in the AGT gene, which means that rather than forming glycine, everything's shunted to oxalate. The problem with oxalate is it's non-soluble, and so it accumulates in the kidneys, causing kidney stones and subsequent renal failure in patients in their early adulthood. And the current therapeutic options, there is an approved siRNA from Alnylam targeting the HAO1 gene. So putting a block in the HAO1 gene means you accumulate glycolate, which is soluble, as opposed to oxalate, which is insoluble. And so this is a perfect indication because there's a clear genetic link, there's a clear biology that the knockdown will work therapeutically, and it allows us to really compare ourselves to an existing therapeutic. Now, the opportunity in the US and EU is between 1,000 and 2,000 patients. We do know there is a significant cohort as well in the Middle Eastern countries for this. So, I think one of the things that we've been talking a bit about, and I think this is something that all of us in this space can relate to, is because we have to shift the thinking when we're going after diseases where your incident population is very small, and your prevalent population's modest, every time we treat a patient, your prevalent population comes down. So we have to think very differently about these diseases as opposed to diseases where you have a relatively robust incident population and it looks more like a traditional therapeutic. And so, we spend a lot of time with some of our investors and with analysts helping them think through, there are two different really types of indications, I think, for these one-and-done therapies, and you have to think about them very differently how you think about the commercial model. So to create the therapeutic, we need to have three things. We need to have guides that work extremely well, that have very high on-target and very low off-targets and are very potent. We need to have a nuclease that functions well, and we need to have an LNP that is potent and works very well in non-human primates. So first step was to identify the appropriate guides. You can see we've got, on the left-hand side, guides targeting either HAO1 or LDHA that have very high on-targets, and the data is not here, but they have extremely low off-target assessments. We also need potent guides. You can see here the HAO1 target 1 guide is much more potent than target 2, so making sure we select guides that have different features and characteristics that enable us to really have an optimal therapeutic. Then we need to go to the mice, and in this particular case, unfortunately, the mouse sequence is very different from human, so these are using mouse guides. But really, do we have a LNP that is potent, and do we have a nuclease that works well? So you see in the left-hand side a very nice dose dependency in the editing of the HAO1 gene in the liver, and you can see it tops out at around 60-ish%, and that's typical because that's about roughly the percent of the hepatocytes within the liver. Some mice are up to 70, some are in 60, but you really can see you've saturated the liver. The other important piece, having the biomarker where we can look for the buildup of glycolate, you can see that in a nice dose-dependent manner as well. You see the glycolate come up significantly in a dose-dependent manner. We can then turn to, there's an AGX mouse model, a knockout model that is an okay mimic of the human disease. And again, you can see in the top left there, we can see the indels in a nice dose-dependent manner very robustly. And we can then look at the activity of the enzyme for the HAO1, which is glycolate oxidase, the GO there. And you can see we knock it down in a nice dose-dependent manner to effectively 0. In the mouse model, we can also look at the urinary oxalate levels, and you can see those fall significantly quickly. So this whole data package gives us confidence to then move into NHP studies, which we have done so, and we're in our running through these 3 different studies. One is to select the optimal LNP formulation. Second study is to identify the dose ranging. The third study to optimize your guide to mRNA ratios. Very standard stuff as we move towards that filing. And just quickly on the discovery engine. So, the value of having this ability to look at the billions and billions of proteins out there is amazing. The downside is that's a lot of data. So we've got a very large computational group of experts where we feel like it sort of brings sort of the best of Silicon Valley with the best of Boston Biotech together to really assess and look at this large data set, be able to pull out the right things that look interesting and bring them into our high throughput screening platform. So, we found tens of thousands of unique nucleases. Now, we're not stamp collecting here, and so we need to identify what are the ones that actually have characteristics we like and which are the ones that we can then utilize to engineer. And you can see in the phylogenetic tree there, we've discovered whole new families. Each one of these dots can be hundreds to thousands of unique nucleases. And we then need to select those that are interesting and bring them into our engineering platform. What you can see here, a typical nuclease, actually, wild-type nucleases really don't function very well in mammalian cells, and so there has to be an engineering approach. And we take a very structure-based approach here, a page from, I think, our drug delivery, from the small molecule world, where we can use structure-based approaches to really generate SAR and know where you're mutating, which allows you to see massive benefits in the editing output. We do have multiple partnerships. Vertex, we have 2 in vivo partnerships there. The first in vivo partnership with Vertex was looking at 5 indications. We just expanded that in January to also include reverse transcriptase-based editing. And then we have multiple ex vivo partnerships which allow folks to access our technology for their particular cell therapy. A team is important. We're very proud of the team we have, and in the interest of time, I won't go into the details, but a team that knows how to translate these therapies, these novel technology into therapeutics. As we think about the future, it's really around execution and moving our pipeline forward, at the same time making sure we continue to innovate on the discovery side, because this field is moving quickly and we want to make sure we continue to be at that cutting edge of it. So that's an important piece for us. And with that, I will see the little timer just ran out, so I will pause to take any questions there may be. Thank you. All right, if there's no questions, that means I overwhelmed you with amazing data, so thank you. All right, thanks.